Hello students, welcome to SK Biology. Today we are going to study a very important topic from molecular biology, bacterial DNA replication. E. coli DNA replication is the process by which bacterial cell duplicates its genetic material before cell division by binary fission as shown in figure. E. coli DNA replication has three unique properties. First one is it is semi-conservative. As proved by Michelson and Stahl, the parental double-stranded DNA separates and acts as a template for synthesis of new DNA. So, new DNA formed will be having one old strand and one new strand. Second feature is E. coli DNA replication is bidirectional, where DNA synthesis occurs in two directions, one towards replication fork and one away from replication fork. Third is location of DNA replication. In E. coli, DNA replication is carried out in cytoplasm. E. coli is prokaryotic organism and it lacks true nucleus. E. coli contains single circular double-stranded DNA with one origin of replication site. This origin of replication contains AT rich region, that is adenine thymine rich region, along with DNA boxes. Now the question is, why AT rich region? AT base pairs contains two hydrogen bonds, so they can be easily opened. Once origin of replication is set for replication, the first step of replication is initiation of replication. Firstly, DNA A protein binds to DNA A boxes at ori C and DNA helix starts opening. DNA C helps to load DNA B, that is helicase enzyme. It breaks hydrogen bond and unwinds DNA at ori C. To maintain single-stranded state of ori C, single-strand binding protein, that is SSB, binds to single-stranded DNA at origin and avoid rejoining of these two single strands. DNA gyrase removes supercoiling ahead of replication fork. Once the opening of ori C is stable, now it comes to an next important step, that is elongation. DNA polymerase cannot start synthesis on its own. So, it requires primase enzyme, that is DNA G, which provides short RNA primer. Then main enzyme DNA polymerase III functions. It has two main activities. One is DNA synthesis in 5' to 3' direction and another is proofreading, that is exonuclease activity in 3' to 5' direction, which corrects mistake during nucleotide addition. During elongation, DNA synthesis occurs in two directions. One towards fork, called as a leading strand synthesis or continuous strand synthesis. It requires only one primer. And the second direction is away from fork, which is lagging strand synthesis. This forms small Okazaki fragments in the size of 1000 to 2000 nucleotide. This strand synthesis is discontinuous and required multiple primers. After completion of synthesis, DNA polymerase I plays important role. It removes RNA primer by 5' to 3' exonuclease activity. Then another enzyme, ligase, fills gap between Okazaki fragments and forms phosphodiester bond. Then replication process comes to an end, where termination of replication starts. TUS protein binds at TER site, that is termination sites, and stop replication fork movement. It gives rise to two identical circular DNA. Final separation of these circular DNA is done by topoisomerase 4. Bacterial DNA replication is highly accurate because DNA polymerase has proofreading activity and it corrects error immediately. The chances of mistake is 1 mistake per 10 raised to 3 to 10 raised to 10 nucleotide addition. So here we completed the important point DNA replication in bacteria.